You know, that weird tingling when your foot falls asleep,
Hmm.
and then that rush of feeling when the blood comes back?
Yeah, pins and needles.
Right. So let's unpack that for a second. Now imagine that same thing, blood flow cut off, then returning, but happening to your heart or your kidneys, maybe your limbs, vital organs.
That's essentially what we're digging into today. It's called Ischemia Reperfusion Injury, or IRI for short.
IRI. Got it.
And for you listening, maybe you're prepping for a discussion, or perhaps you're just, curious about how the body handles this kind of stress.
Or maybe you've heard the term thrown around.
Exactly. Our goal today is really to break down this complex process. We've been through the source material. It's a PDF titled 006 Ischemia Reperfusion.
Right.
And we've tried to pull out the really key insights. The mission, if you like, is to understand this kind of two-stage problem with IRI. First, the lack of blood flow, the ischemia.
The starving of the tissues.
Precisely. And then the reperfusion, the return of blood flow, which kind of paradoxically can actually cause more damage. We want to explore what that really means.
Okay, so it's not just a simple summary we're doing here. We're looking for the important bits, the surprising parts. The source uses this analogy, like a 1-2 punch.
Yeah, I like that analogy. The first punch is the ischemia itself.
The initial hit.
That's it. Blood flow is cut off or at least drastically reduced. Cells get stunned, they're starved of oxygen, nutrients, everything they need.
But they don't just give up immediately, do they?
No, interestingly, they have some initial resilience. There are these preconditioning pathways that can kick in, almost like a cellular emergency defense.
A built-in survival mode?
Kind of, yeah. But, and this is crucial, it's time sensitive. As the source makes really clear, if that lack of blood goes on too long,
Those defenses get overwhelmed.
right, and the cells start heading towards irreversible damage, cell death, which is why, the speed of intervention, like from a vascular surgeon, is so critical.
Because their job is to get that blood flowing again, ASAP.
Exactly. Limit that ischemia time.
Which brings us to the second punch, reperfusion. It sounds like the solution, the rescue.
It sounds like the good news, doesn't it? But it's the paradox. Restoring the blood flow, which you absolutely have to do, can trigger this massive surge of signaling molecules. Chemokines?
Chemokines, like distress signals.
Sort of, yeah. They're released by the already damaged cells and also by immune cells rushing to the scene. And this chemokine storm causes even more damage.
Not just locally.
No, that's the scary part. It can cause local damage, sure, but also potentially systemic problems affecting other parts of the body. It activates both main pathways of cell death necrosis and apoptosis.
Which can lead to organ failure.
In severe cases, yes. It can be really serious, even fatal.
Wow. Okay, so it's this incredibly tricky balance. You need reperfusion, but it comes with its own risks. The source mentions, going back a bit historically, to this thing called the no reflow phenomenon. What was that about?
Yeah, the no reflow idea came from early research, especially on heart attacks. Researchers found that after a long period of ischemia, even if you cleared the main blockage in the artery,
The blood still wouldn't flow properly.
exactly, the smallest blood vessels, the microvasculature, the capillaries, they themselves became so damaged or blocked that they just couldn't accept the blood flow.
Like the downstream pipes are clogged or broken?
That's a good way to put it. And obviously that's a massive problem. If the tissue downstream can't get blood, it can't recover, even if the main problem is fixed.
That sounds dire. But then almost the opposite was discovered, ischemic preconditioning.
Right. It seems totally counterintuitive. Short bursts of ischemia, followed by reperfusion, can actually protect the tissue against a later, longer ischemic event.
How on earth does that work? Stressing it out makes it stronger.
Kind of, yeah. It seems these brief stress periods trigger protective mechanisms inside the cells. The source talks about two windows of protection.
Windows.
Yeah, like time frames. There's a first window that happens pretty quickly, lasts maybe up to 2 hours. Then there's a second window, which is delayed, starts around 24 hours later and can last up to 72 hours.
So immediate and longer-term protection.
Exactly. And that second window seems to involve changes in which genes are active. The cells start making more protective stuff, things like heat shock proteins, antioxidant enzymes, and inducible nitric oxide synthase.
So the body adapts to the stress.
It seems so. It suggests there are both immediate and longer-term adaptive responses. Understanding how these different phases get triggered could be really key for developing therapies, mimic that protection.
Fascinating. And more recently, the understanding shifted slightly. It's not just the cells, but the blood vessels themselves that get damaged structurally by IRI.
Yes, that structural damage to the vasculature is now seen as really critical. It's not just about flow and cells reacting. The actual pipes, the blood vessel walls, get compromised.
Which makes fixing things even harder.
Absolutely. It brings us back to that challenge the source mentions, finding the Goldilocks recipe for reperfusion. Not too much, not too little, just right timing and approach. And honestly,
Okay.
we're still working on that. It remains a huge hurdle.
A real balancing act. Okay, let's dive deeper into the vasculature itself. The source calls it a microscopic battlefield. What happens to the endothelium first? The inner lining.
Right, the endothelium.
Yeah.
Normally, it's this smooth, anti-clotting barrier, but it's one of the first things hit by IRI. During ischemia, the lack of oxygen means no energy, right? ATP depletion.
The cell's fuel runs out.
Exactly. This messes up the cell's electrical balance, causes stress. It can even kill the endothelial cells directly. Then, when reperfusion happens, all those inflammatory signals get released.
The chemokine surge we mentioned.
Yeah, and that disrupts the connections between the endothelial cells, the tight junctions and adherens junctions. Think of them like the mortar between bricks.
So the wall becomes leaky.
Precisely. It increases permeability, fluid leaks out, proteins leak out. That causes swelling, edema, and it lets inflammatory signals spread more easily. Plus, the glycocalyx gets damaged.
So what now?
The glycocalyx, it's like this slippery protective coating on the inside of the blood vessel. IRI damages that too.
Okay, so leakiness, swelling. What else?
Well, during reperfusion, the production of nitric oxide, or NO, goes down. NO is crucial for relaxing blood vessels and preventing clots.
Less relaxation, more constriction.
You got it. And at the same time, the production of damaging molecules, those reactive oxygen species, or ROS, goes way up. So the endothelium gets pushed into this activated, pro-clotting, pro-inflammatory state.
A perfect storm inside the blood vessel. What about the next level down, the arterioles, the smaller arteries?
Similar problems, really. IRI messes with their ability to relax properly in response to nitric oxide. They become more prone to constricting instead.
So they clamp down.
Right. The source points to a key study from way back in 1987 that really nailed the importance of nitric oxide in this impaired relaxation during IRI.
And why does NO production fail?
Well, endothelial cells have different ways of making NO. One is always on, kind of. But during IRI, an enzyme called arginase gets hyperactive.
Arginase.
Yeah, and it chews up L-arginine, which is the exact molecule you need to make nitric oxide. So the raw material runs out just when you need it most.
So you can't make this stuff that keeps the vessels open.
Exactly. And this impaired relaxation also affects the smooth muscle in the vessel walls, making them constrict even more. You get sluggish flow, reduced forces inside the vessels. It's a vicious cycle of poor perfusion that can spread systemically.
Okay, down to the tiniest vessels near the capillaries. The source says the dysfunction there is occlusive. What does that mean? Blocked.
Yeah, essentially blocked. It happens in a couple of ways. Either the capillaries themselves physically narrow down,
How?
often because of swelling, edema in the tissue around them, squeezing them shut, or they get plugged up by white blood cells, leukocytes.
White blood cells get stuck.
They do. Remember those activated endothelial cells? They become sticky. Platelets stick. Leukocytes stick. And leukocytes aren't very flexible. They're quite rigid. So they can literally jam up these tiny, narrow capillaries, like a microscopic traffic jam.
Can anything be done about that?
Well, some interesting studies in mice. Mice engineered to lack certain adhesion molecules. The Velcro that helps cells stick seem to be somewhat protected.
Less sticking, less plugging.
Seems like it. There's also this thing called stall dynamy, basically neutrophils blocking capillaries, which can sometimes be improved with antibodies against those neutrophils. And boosting antioxidant enzymes like superoxide dismutase has helped capillary flow in animal models too.
And this capillary blockage is the main reason for that no reflow phenomenon.
That's the current thinking, yes. It seems to be the origin of why blood can't get through even after the main blockage is gone.
Okay, makes sense. Now, what about the venules? The small veins carrying blood away. You said the inflammation really kicks off there.
Yeah, the venules seem to be ground zero for the peak inflammatory response. Again, that slippery glycocalyx lining gets damaged early on.
Making it easier for cells to get out.
Exactly. It promotes neutrophil extravasation, that's neutrophils squeezing out of the bloodstream. Enzymes like xanthine oxidase and MMPs help this along.
MMPs?
Matrix metalloproteinases, enzymes that break down tissue structure.
Gotcha.
And then immune cells already living in the tissue, like mast cells and macrophages, get activated by the whole situation. They move towards the venule lining, releasing signals that call in more leukocytes, making the venules even leakier.
Or chaos.
Pretty much. Then platelets start sticking together, forming little clots. And all this contributes to that systemic inflammation, fluid buildup, maybe even problems with blood pressure.
So it's a coordinated attack. And neutrophils seem key. The source calls it an innate immune assault. What's their main weapon?
Their main weapon, probably those reactive oxygen species, ROS. Activated neutrophils churn them out using enzymes like NADPH oxidase and myeloperoxidase.
What kind of ROS?
Things like superoxide and also hypochlorous acid, which is basically bleach, really damaging stuff.
Bleach, wow.
Yeah. Plus, they release enzymes that degrade tissues, those MMPs again. They really chew up the structural proteins. And this whole process, because there's no infection involved, is called sterile inflammation.
Sterile inflammation.
Yeah.
But it's still causing damage.
Absolutely. It's essential for normal healing, but in IRI, it just goes overboard and drives more injury.
And this movement of neutrophils out of the vessels, the transendothelial migration, that ramps up during reperfusion.
Yes, precisely. When oxygen and nutrients return, that's when this coordinated movement really peaks. You get more fluid leakage, too. The endothelial cells put up more sticky molecules, ICAM1, VCAM1, E-selectin.
Like Velcro again.
Exactly. So neutrophils can grab on, roll along the vessel wall, stick tight, and then squeeze through the gaps into the damaged tissue. Chemical signals like TNF-alpha and IL-1 then guide them to the exact spot.
Like little guided missiles. And they release stuff from granules, too.
They do. Those granules are packed with more weapons. Myeloperoxidase, which helps make that bleach-like acid. Elastase, other proteases like MMP8 that degrade the tissue matrix. These things cause direct damage, make membranes leaky.
And this can even affect organs far away.
That's the concerning part, yes. There's evidence linking neutrophils activated by IRI, say after aortic clamping, to injury in distant organs. It highlights the systemic nature of this response.
It really is a cascade. Okay, let's shift gears and look at some specific clinical scenarios. Heart attacks first, myocardial stunning, and hibernation. How does IRI play out there?
Oh, it's hugely important in cardiology. After procedures to open blocked heart arteries, whether it's clot-busting drugs or angioplasty and stenting or even bypass surgery.
Thrombolysis, PCI, CEBG?
Right. Reperfusion is essential, but it brings IRI. This can cause arrhythmias, those irregular heartbeats. It can cause myocardial stunning, where the heart muscle is temporarily weakened, and it can cause actual heart muscle cell death, myocyte death.
Why the arrhythmias?
It seems that after prolonged ischemia, the inner membranes of the mitochondria, the cell's powerhouses, lose their stability. This affects the electrical balance needed for a normal heartbeat.
And a lot of the heart attack damage might actually be from the reperfusion itself.
That's what the evidence suggests. A significant chunk of the final infarct size, the area of dead tissue, might be attributable to the injury caused by restoring blood flow. Muscle that was alive right after reperfusion might die hours later because of IRI.
Which leads to stunning, that temporary weakness.
Exactly. Stunning is thought to be this kind of protective, maybe adaptive response by the heart muscle. It's reversible. But importantly, for it to be called stunning, the blood flow has to be pretty much back to normal.
How's that different from hibernation?
Hibernation is when the heart muscle function is reduced, but the blood flow itself is also chronically reduced. Stunning is temporary weakness with good blood flow. Hibernation is sustained weakness with poor blood flow, waiting for restoration.
But both involve the heart muscle sort of powering down.
In a way, yes. Physiologically, stunning is linked to oxidative stress, calcium overload inside the cells, makes them less responsive. Interestingly, both stunning and hibernation seem to trigger genes that promote cell survival.
And hibernation is linked back to that preconditioning idea.
It is, yeah. The idea that repeated short stresses adapt the heart muscle is very similar to ischemic preconditioning with those first and second windows of protection we talked about.
Has this led to treatment ideas? Trying to mimic preconditioning?
Definitely. Lots of research. The source mentions the DNMI3 trial looking at post-conditioning, applying brief ischemia after blood flow is restored, or delaying stenting slightly to see if that helps during PCI.
What about that remote preconditioning, like squeezing an arm to protect the heart? RIPC.
Yeah, RIPC. It's shown some promise, particularly in reducing markers of heart damage during bypass surgery, but the results on major outcomes like death or major heart events have been mixed.
Not a home run yet.
Not consistently, no. It might depend on the type of surgery, the patient, and it really seems like the duration of the initial ischemia is still the biggest factor driving how much IRI you get.
Any other approaches? Drugs.
Yes. Trying to infuse protective drugs is another strategy. Adenosine, for instance. The MSDOD trials showed it could reduce infarct size in certain heart attack patients.
How does adenosine help?
It's thought to help relax blood vessels after reperfusion and maybe dampen down some of that inflammation in the heart tissue.
Okay, let's switch to limbs. Acute limb ischemia, ALI. What happens when blood flow is cut off to an arm or leg and then comes back?
ALI is usually from a sudden clot. When reperfusion happens, you can get massive muscle swelling in the limb. This releases a lot of stuff into the blood myoglobin from damaged muscle, potassium.
Which can cause problems elsewhere.
Big problems. That myoglobin can damage the kidneys, cause acute renal failure. The potassium can affect the heart, and the swelling itself can be dangerous.
Compartment syndrome.
Exactly, it can lead to that. Sometimes surgeons have to do fasciotomies, cut the tough tissue surrounding the muscle compartments to relieve the pressure. But even that might not save the muscle, and in the worst cases, amputation is needed.
Even if it's not that severe immediately.
Even then, a lingering inflammatory response can cause chronic problems and potentially lead to amputation down the line.
What about chronic limb ischemia, where flow is poor for a long time? Does IRI matter when they try to fix it?
It does. When they do procedures to improve blood flow, the reperfusion can still cause IRI, mainly through oxidative stress-damaging muscle cells and their mitochondria.
Have treatments work there? Antioxidants.
Some preclinical studies showed modest benefits with antioxidants or NO donors, but it hasn't translated clearly into major clinical benefits for patients yet. Right now, the main treatment for ALI is still anticoagulation, preventing more clots.
And attempts to control the reperfusion itself haven't panned out.
There was a trial called SURAIL that looked at controlled reperfusion, but unfortunately it didn't show a significant benefit in preventing amputation or death. Still a challenging area.
Speaking of compartment syndrome, you mentioned it can happen in ALI. How does IRI drive that specific condition?
Right, so compartment syndrome is all about pressure buildup in a closed space, usually muscle wrapped in fascia. The initial injury or swelling increases pressure.
Which squeezes the blood vessels.
Exactly. Microvascular compromise. Less blood flow, less oxygen, pH drops. Then the leakiness from IRI causes more fluid buildup, raising the pressure even more. Less oxygen again. It's that vicious cycle.
Leading to muscle death.
Muscle death, nerve damage, kidney failure from the muscle breakdown products. It's really serious.
And the treatment is that emergency fasciotomy.
Yes, and time is absolutely critical. There's this idea of a two-hit inflammation model. The first hit is the initial injury and ischemia. The second, potentially worse hit, comes after the fasciotomy when all that damaged cell debris and inflammatory stuff floods out into the general circulation.
So delaying the fasciotomy is really bad.
Very bad. Significantly increases amputation risk. The anterior compartment of the lower leg is especially vulnerable because a key nerve runs through it.
How quickly does nerve damage happen?
It can start within an hour and become irreversible after maybe 6 hours. It's a surgical emergency.
A very tight window. Okay, another scenario. Clamping the aorta during surgery. That causes IRI too.
Absolutely. When you clamp the body's main artery, the tissues downstream are ischemic. When you release the clamp, reperfusion injury happens.
What does clamping do to the body's overall circulation?
Initially, clamping increases systemic vascular resistance, the resistance the heart pumps against. The body releases things that raise it further. Then, when you unclamp, resistance drops suddenly.
Why?
Partly because the vessels below the clamp dilate, and partly because vasodilators like adenosine and lactate built up during ischemia get released. This drop can actually worsen the IRI. Surgeons have to manage this carefully, unclamping slowly, giving fluids.
And the higher the clamp, or the longer it's on.
The greater the risk of IRI, yes, and also a risk of spinal cord injury, depending on where the clamp is. Sometimes they use hyperthermia, cooling the body, to try and protect the spinal cord.
Is IRI different in nerves versus muscles?
The basic pathophysiology is similar, but neurons are generally much more sensitive to oxygen deprivation. A lot of research focuses on making neurons more resilient. Newer, less invasive endovascular techniques help by reducing clamp time. And some drugs, PRP1 inhibitors, look promising in early studies.
What about the gut? Mesenteric ischemia.
The intestines are really vulnerable to IRI. Huge surface area, plus that whole microbiome, all the bacteria living there.
Does reperfusion cause more damage there too?
Yes. Classic studies by Parks and Granger showed that reperfusion after intestinal ischemia can actually be more damaging than the ischemia itself.
Why is the gut so sensitive?
During ischemia, the gut barrier breaks down. Bacteria can leak out, cells run out of ATP, mitochondria get damaged. If it goes on too long, cells die via necrosis. Then with reperfusion, you get the ROS burst, immune cell recruitment.
Same pattern.
Same pattern, but amplified by the gut environment. The complement system gets activated, inflammatory cytokines are released. Even specialized gut cells, called Paneth cells, which normally fight microbes, become dysfunctional. It really compromises the barrier integrity.
And this relates to hemorrhagic shock too. Massive blood loss.
Yes. The global ischemia from shock, followed by giving large volumes of fluid resuscitation, can cause massive intestinal edema, swelling. This can lead to abdominal compartment syndrome pressure buildup in the abdomen itself, further restricting blood flow.
Requiring surgery to decompress.
Often, yes. Techniques like Reboe, using a balloon to temporarily block the aorta internally, can help control bleeding, but the downstream ischemia they cause is still a major limitation.
And the worst case systemic outcome is MODS. Multi-organ dysfunction syndrome.
MODS is devastating. It's driven by that uncontrolled chemokine storm from widespread IRI. It's a leading cause of death in the ICU.
How does it typically progress?
Often starts with the lungs acute respiratory distress syndrome, ARDS, then liver failure, kidney failure, GI dysfunction. The lungs seem particularly vulnerable to the inflammatory mediators released from the gut and liver after ischemia.
And it involves clotting issues too.
Yes. It's a complex mess. Thrombosis, DIC, disseminated intravascular coagulation. It's multiple organ systems failing due to this runaway inflammation. A really grim outcome.
But there was a slightly more hopeful note. Younger people handle IRI better.
Yeah, there's definitely evidence, both anecdotal and experimental, suggesting younger patients tolerate IRI better.
Why might that be?
Well, they generally don't have the underlying conditions like vascular disease or diabetes that worsen IRI. Understanding why they're more resilient, what protective pathways they have, could be really valuable for developing therapies for older adults.
And conversely, things like diabetes make IRI worse.
They do. Diabetes seems to accelerate cellular aging processes, making tissues more vulnerable to IRI through various molecular pathways.
Interesting. Okay, let's get right down to the cellular level now. During ischemia itself, before reperfusion, what's happening inside the cell?
Okay, so oxygen is cut off, first thing. Metabolism goes haywire. ATP, the cell's energy currency, plummets. The cell desperately switches to anaerobic metabolism, glycolysis.
Which produces lactic acid.
Right. So the inside of the cell becomes acidic, the pH drops. This activates a pump on the membrane, the sodium-hydrogen exchanger. Sodium rushes in.
And water follows sodium.
Exactly. The cell swells up, hydropic swelling. Meanwhile, enzymes that need ATP stop working. The pumps that normally kick calcium out of the cell or stash it away fail.
So calcium builds up inside.
Yes, intracellular calcium overload. This activates damaging enzymes called calpains. It also triggers the conversion of xanthine dehydrogenase into xanthine oxidase.
Which becomes a problem later during reperfusion.
Precisely. It gets primed to produce ROS as soon as oxygen returns.
And even with low energy, genes are changing.
Remarkably, yes. Even as overall protein production slows down, the levels of a key protein called HIF-1 alpha shoot up.
Hypoxia-inducible factor.
That's the one.
Yep.
And HIF-1 alpha turns on genes like VEGF, which tries to stimulate new blood vessel growth. Other stress response pathways involving NF-κB, AP1, P53 also get activated, leading to more pro-inflammatory signals.
But the low pH actually slows some of this down.
It does, interestingly. That acidic environment can inhibit some enzymatic processes. It buys the cell a little time, highlighting again why restoring flow quickly but carefully is so important.
Then comes reperfusion. The paradox. Bringing back oxygen is good, but bad.
It's the double-edged sword again, yes. Oxygen comes back, ATP production restarts, pH normalizes, which is all needed. But T, this sudden influx of oxygen, fuels a massive burst of reactive oxygen species, ROS.
And where does this ROS burst come from mainly?
Primarily from the mitochondria, the powerhouses. As they ramp up energy production again with oxygen, the electron transport chain kind of leaks electrons, generating ROS.
Yeah.
And this ROS flood then triggers more cell death, both apoptosis and necrosis.
Okay, let's break those down. Apoptosis first. That's the programmed, tidy death.
Right. Think of it as cellular self-destruction, gene-directed. In IRI, it's triggered by both the hypoxia and the oxidative stress of reperfusion. It's mainly controlled via the mitochondria, involving the BCL2 family of proteins.
How does it start?
The main route in IRI is the intrinsic or mitochondrial pathway. Proteins like BAX and BAT get activated, and they essentially poke holes in the mitochondrial outer membrane.
Making it leaky.
Exactly. Stuff leaks out, including cytochrome C. Cytochrome C then joins up with other factors to form the apoptosis.
It's ominous.
It is for the cell. The apoptosis activates executioner enzymes called casposes, and the casposes then go about dismantling the cell in an orderly fashion.
What does an apoptotic cell look like?
It shrinks, the chromatin condenses, DNA gets chopped up, the membrane bulges out in blebs, and eventually it breaks into neat little packages called apoptotic bodies. These get cleaned up by other cells without causing much inflammation. A key eat-me signal is phosphatidylserine flipping to the outside of the membrane.
So relatively clean. What about necrosis and this other one, necroptosis?
Necrosis used to be seen as just messy, uncontrolled cell death, swelling, bursting, spilling contents causing inflammation. But now we know about necroptosis, which is actually a regulated form of necrosis.
How's it regulated?
It can be triggered by some of the same signals as apoptosis, like death receptors, but it follows a different internal pathway involving proteins like RIP kinases. It requires calcium and ROS. A key event is the opening of a big pore in the inner mitochondrial membrane, the MPTP.
Mitochondrial permeability transition pore.
That's it. When that opens, the mitochondrion loses its charge, can't make ATP properly, swells up, and releases damaging factors. The cell loses essential molecules like NAD plus A and eventually lyses, releasing its contents and triggering inflammation.
But you said necroptosis pathways might be druggable.
Yes, because it's a regulated pathway, unlike random necrosis, there are specific molecular steps that could potentially be targeted with drugs. The idea is maybe you could block necroptosis to keep tissues in a more dormant, protected state during ischemia, kind of like hibernation. It's a really promising area.
Very interesting. Okay, moving to circulating factors.
Yeah.
ROS, again, how do they spread their damage?
So when reperfusion happens, that xanthine oxidase we mentioned gets to work, producing superoxide and hydroxyl radicals. These don't just stay put. They can travel systemically.
Hurting endothelial cells everywhere.
Potentially, yes. Hydroxyl radicals are particularly nasty. They cause lipid peroxidation, damaging cell membranes, and releasing pro-inflammatory fats. Xanthine oxidase derivatives also make endothelial cells stickier and attract more neutrophils. It contributes to that chronic oxidative stress seen in conditions like peripheral artery disease.
What about the complement system? How does that immune pathway get involved?
Complement is part of our innate immunity, a cascade of proteins that can ultimately form the membrane attack complex, or MAC.
Which punches holes in cells.
Exactly, leading to lysis and death. We've known since the 70s that complement gets activated in IRI, initially found in ischemic hearts. Levels of complement proteins go up in ischemic tissues.
How does it get activated?
It seems changes in the cell's internal structure during ischemia can expose new targets on the surface that complement binds to. While blocking some complement receptors hasn't worked well in human trials, targeting the MSC components directly, like C5 or C9, with drugs like eculizumab still holds potential.
And neutrophils contribute more than just ROS. They have enzymes too.
Yes, that neutrophil-mediated tissue injury, or NMTI. They release proteases, enzymes like cathepsin G, elastase, that directly break down tissue components, contributing to necrosis and loss of function. And of course, there's that whole flood of chemokines orchestrating the chaos, though the source notes much of that work focused on organs like the liver.
Such a complex web. Okay, finally, where's the research heading? What looks promising for the future?
Well, as we mentioned, those necroptosis pathways are a hot area. There's this balance between caspase 8, which inhibits necroptosis, and RIP3, which promotes it. Targeting that balance point could be key.
Finding a way to tip the balance towards survival.
Or at least away from inflammatory death. Mice lacking both apoptosis and necroptosis pathways are surprisingly resistant to inflammatory injury, which is telling. Another big area is figuring out how to modulate the immune response itself. Maybe harness inflammatory cells to limit the damage rather than cause it.
And that table in the source mentioned a few other potential targets.
Right, just briefly. Things like endothelin, a vasoconstrictor blocking it helps in animal models. Adenosine, we talked about its protective effects. Adrenomegalin, another vasodilator. The heme oxygenase system and its product, carbon monoxide, have anti-inflammatory effects. Anticoagulants are already standard practice. Hypothermia can be protective. Even controlling nerve signals, like through the vagus nerve, shows promise in animal models. Lots of avenues being explored.
Sounds like it. Okay, so let's try and synthesize this. What's the big takeaway message about IRI?
I think the core message is just how complex IRI is. It's this two-act play. The ischemia causes initial damage, but the reperfusion, while absolutely essential, unleashes the second wave of injury through inflammation, ROS, cell death pathways. It's this incredibly delicate balance between rescue and further harm.
Right, a real tightrope walk for clinicians.
Right.
And for you listening, whether you were deep diving for a specific reason like understanding vascular disease better, or maybe just because the body's response to stress is fascinating.
Which it absolutely is.
Hopefully this discussion has given you a clearer picture of the microscopic battlefield, the key players,